Well, thanks for having us back again this year. It's nice to be able to give you an update on where we are. We've made some significant progress in the last year and in the last few months in particular. For those of you that don't know us, we are a UAMS Arkansas BioVentures company. We were formed to develop and promote UAMS developed technology, and the reason we do it this way is so we can facilitate both high-quality science and efficient business practices. The vision of our company is to be a leader in antidote-type therapies, particularly, and our mission is to develop new medicines for the treatment of disease, specifically medicines that are biologic in origin. The diseases that we have targeted to start with are those of substance abuse, both long-term recurring illnesses and short-term acute illnesses. Addiction in particular can be characterized as a long-term recurring illness like many other diseases such as diabetes. Addiction has physiologic derangements. It can be treated with medicines, but it also requires lifestyle changes. This is very similar to the treatment of diabetes. We use medicines like insulin to treat the physiologic derangements, but it requires lifestyle changes on the part of the individual in order to actually get better over time and to have a good outcome. Our initial focus is on the stimulants methamphetamine and methamphetamine-related drugs like amphetamine and ecstasy. Now, if you look at the needs over the past several years for medicines of this type, you might think that to a degree there's been some decrease in the numbers of people looking for treatment, and indeed the numbers who sought treatment for meth abuse and meth addiction in 2009 was down to 110,000 from a high reported about 3 years ago of 160 or 170,000. Similarly, the numbers of ER visits specifically related to methamphetamine are down to around 125,000. If you look at national statistics on people who used methamphetamine in the last month, currently they're reporting about 350 to 360,000 in the last month. So you could say there's only 300 to 400,000 regular users. Well, I'm going to make an argument that there's a few more than that currently in the United States and abroad. If you look at worldwide numbers for regular methamphetamine abuse, you see numbers that suggest that there's 10 to 20 million people worldwide who are regular methamphetamine users. So what we see here and what we believe is the numbers of users and the people who are seeking treatment are actually underestimated, and there's 2 or 3 reasons for that. One is that this population is not going to be real prone to reporting their use. That's pretty much a given. 2, the reporting statistics that we get from the federal government are only those from publicly funded treatment centers. We think that that only represents maybe a third of the numbers of treatment centers nationwide. The biggest reason I think that we're seeing a decrease in perhaps the numbers of people seeking treatment is that there's a perception out there among users that current treatments, such as they are not effective. I saw a recent survey that suggested that 90% of people don't get help because they don't think it will work. The same survey suggested that 25% of people don't get help because of expense or because of cost of the medicines, what medicines that are available. So one of the things we anticipate is that the availability of a medicine will increase the need for that medicine. Again, to go back to my diabetes analogy, the diagnosis of diabetes changed dramatically with the advent of insulin and with the improved insulins over time. The blood sugar at which you diagnose diabetes came down from around 300 to currently at about 140 because of the advent of better medicines. So basically, the numbers of diabetics may not have gone up, but the numbers of people being treated for diabetes went up because of the advent of an improvement in the medications. The other reason that we think our medicine is important is there are no approved medicines for the treatment of methamphetamine abuse. None approved by the FDA. So what are our medicines? We make our medicines as antibodies. These are basically the same molecules that you have in your body to fight infections, to fight other foreign pathogens that get into your body. So they're naturally occurring molecules. These medicines, antibodies, once we give them to you, are there to attach to and neutralize the medicine or the drug, methamphetamine in this case. The nice thing about what we're doing right now is that there are no medicines for the treatment of drug abuse on the market. There are no antibodies for the treatment of drug abuse, particularly methamphetamine, but there are medicines, antibody-based medicines available for cancer, autoimmune diseases, infectious diseases, and even prevention of transplant rejection. So the FDA has a great deal of experience with monoclonal antibodies. There are about 22 or 23 currently on the market at this point. We also have vaccines, active vaccines, where we give you a shot, like a tetanus shot, and you develop your own antibodies against methamphetamine. This project is not quite as far along as the monoclonal antibody project, but I'll tell you more about that as we get along here in the talk. This technology was all developed at UAMS by Dr. Michael Owens, my collaborator of the past 18 years. Our goal is to target the drugs themselves and not the receptors, and I'll explain more about that in just a minute. So how do our medicines work? Well, I've shown here a cartoon of a brain. Let's put a magnifying glass on it so you can see the blood vessels and the nerves in the brain. So the blood vessels are here in red. My arrow showing. Yeah. The nerves are the blue, and then the methamphetamine or drug of abuse is out here in yellow. You can see that when the drug of abuse or methamphetamine is taken into the bloodstream, it rapidly and very extensively crosses into the brain where it exerts its action. There's basically no hindrance for methamphetamine crossing from the blood vessels into its sites of action in the brain. Our medicine, an antibody, is a big molecule, and it essentially all stays out in the bloodstream. It is stickier, as it were, for the methamphetamine than methamphetamine sites of action. So what happens is the antibody basically sets up an equilibrium change and draws the drug of abuse of the methamphetamine back out into the bloodstream from whence it can be eliminated from the body. So very straightforward. We target the drug itself and not the receptors in the brain where the drugs can work. So what are our products? We have a monoclonal antibody against methamphetamine, and again, this is the product that is the farthest along in its development. This is a cartoon showing that it's a great big Y-shaped molecule. It has two sites on each molecule for binding of methamphetamine. We also have a vaccine in production and in development, which is, again, not quite as far along, but it's doing quite well in our preclinical studies. We have two potential indications for our medicines. One is prevention of relapse to methamphetamine use for users who seek treatment, who go into therapy, who are able to stop using at least temporarily, and then when they fall off the wagon, which they will, we will prevent the reinforcing effects of methamphetamine and hopefully keep them in treatment for longer periods of time. The longer you keep people in treatment, the better your chances of success. But also, we think one of our indications will be for overdose. There are no antidotes for the treatment of methamphetamine toxicity. Methamphetamine can cause heart attacks, strokes, severe delirium. It can cause all kinds of chronic and acute problems. So if you've got a patient who comes to the emergency room right now who is methamphetamine toxic, the treatment of choice is to put them in 4-point leather restraints, treat them with sedatives until the methamphetamine wears off. So one option would be to give them a small dose of our antibody, reverse the toxic effects of the methamphetamine, and hopefully get them out of the emergency room a little bit quicker. So what is our strategy, our business strategy? It's basically to use the university side to do drug development, to do the preclinical testing, to do the things that we need to do to determine that, at least theoretically, our medicine will be ready for use in humans. We want to outsource and have outsourced our manufacturing, our preclinical toxicology testing, and will outsource our clinical trials. This helps us to reduce our capital needs, but it also helps us to set up a firewall for conflicts of interest. Those of us, there's 4 of us that are primary partners in the company. We all own parts of the company. As the medical doctor, I cannot do the clinical trials for this medicine. That presents a conflict of interest. But I can farm that out to another company. Ultimately, we would like to enter the market after pivotal clinical trials through either collaborative marketing strategy, some kind of licensing or merger and acquisition. And then get back to what we do best, which is to develop new medicines. Our team, as many of you have heard about before, and I've already mentioned, started with Mike and me. Mike is the inventor of this technology. He and I have been working together for nearly 18 years. About 10 years ago, we started collaborating with Dr. Ralph Henry at the University of Arkansas in Fayetteville, who is our vice president for production and biopharmaceutics. About the time we formed the company InterveXion in 2004, we had made contacts with our CEO, Dr. Holtz, and with our Chief Financial Officer, Mr. Jerry Damerow, who has been a tremendous help to us basically on a volunteer basis. A couple of years after we started the company, Dr. Misty Stevens came to us as Operations Director. She is the brains behind the whole operation and keeps us all on track. So with any new medicine, you have risks, and so we're not any different in that respect. First of all, you have to determine that your medicine is safe. Well, we've done a couple of things to design a safe medicine. First of all, we've chosen the type of antibody specifically. Scientifically, it's called an IgG2 antibody. This type of antibody has the lowest risk of causing any kind of untoward effects in humans. And indeed, we've shown already that it doesn't bind some of the proteins that other types of antibodies do bind that could be detrimental. We also do formal toxicology testing. I'll fill you in a little bit more on where we are with that in just a minute. The second risk is it's got to work, or it does you no good. Well, there are multiple antibodies in our pipeline, so if the one that we have chosen to move forward doesn't work, we have others that can move in right behind it. But we have good reason to believe, based on other studies with other antibodies that bind foreign targets, that our medicine should be very effective at what it is designed to do, which is to bind methamphetamine. Nevertheless, we do have contingency plans. Our biggest risk has to do with our market. When we presented here a year ago, one of the goals that we had was to develop and to conduct a market analysis. We do not yet have the money to do that. That is still something that we need to do, and we need to identify the payers, or who will end up paying for this medicine, particularly in the current environment, the healthcare environment that we have. We feel like we have a number of advantages, though, over existing therapies, and I'll show you these therapies one at a time and talk to you about how our medicine fits in and we think has some advantages over these medicines. First of all, there are a number of medicines that have been tried, regular small molecule type medicines, to reduce or prevent the effects of methamphetamine and to help users get off of methamphetamine. The advantage to these medicines is that they're fairly inexpensive because they're all medicines that are currently on the market. None of these, again, have been approved specifically for methamphetamine addiction or toxicity. The biggest problem with these medicines is that they cross that blood-brain barrier that I showed you and interact with receptors in the brain that you need to function normally, because that's where methamphetamine works. And the more these medicines cross and interact with those receptors, the more side effects there are. In particular, and most importantly, the risk of addiction to the treatment medicine. In some cases, that can make somebody functional. For instance, with methadone. Methadone is very good for making heroin addicts functional, but it's still an addiction, so it's not the best solution. Plus, these medicines are not specific for a given drug, like ours would be. The second way you can treat methamphetamine abuse is to put people in jail. Unfortunately, that doesn't seem to work too well because when they come out, they begin using again. Very few people who go in, who are big-time users, come out and say, I'm not going to use anymore. They still keep using. The best available therapy is talk therapy, behavior modification type therapy. It is effective in 15 to 20% maybe of people who undergo it and who stick with the program. Unfortunately, there's a 75% recidivism rate at about 13 months after beginning of therapy, so it's not perfect either. Our medicine, while not perfect, can help in all three arenas. It will not interact detrimentally with other small molecule medications, so if somebody were to come up with something that would be useful, our medicine, because again it doesn't interact with the receptors where these medicines work, can be used in combination with small molecules. There's nothing that says that our medicine couldn't be administered in a criminal justice system or as part of a plea bargain, a plea agreement for somebody to keep them out of jail and to keep keep us from having to pay for their incarceration. Where we think it'll work best is in concert with conventional behavior modification therapies, where you put people into therapy treatment to help them learn lifestyle changes that can help them get over their addiction. We give them an additional protection against their recidivism. If in therapy, they've gotten a dose of our antibody on board, they decide to go out and use, they don't get high because our medicine is on board. They say, I need to go back to therapy, and they can stay in therapy for longer periods of time. We think that's where our medicine fits the best. So let me tell you where we are in our progress. First of all, everything we've done so far has been funded specifically by NIH and the National Institute on Drug Abuse. The meth vaccine has been made into a form that can be suitable for administration into humans. We have developed and validated the testing methodology in animal studies to test it for when we can get the dosing and the immunization schedules optimized. We're still working on that. We have confirmed that this vaccine does generate anti-methamphetamine titers, which means that in the animals they produce antibodies against methamphetamine. We anticipate submitting a new grant in the spring, specifically in March, to fund our trial batch production and toxicology testing so we can be in the preparation mode then for clinical trials, hopefully in about 2 years with this vaccine. More exciting for us is the progress that we've made on our methamphetamine antibody. I've shown here a little arrow that shows us the progress from discovery and optimization out into clinical trials. And just to foreshadow a little bit, we're right there now. How have we gotten there? Well, through years of animal studies and preclinical development, we had developed a number of different antibodies against methamphetamine. We chose one about 3 years ago to take forward and engineered it into a form that is suitable for administration to humans. Through a Recovery Act grant, which we obtained a little over 2 years ago, we're able to get the master cell bank for this medicine produced. We have completed the manufacturing. We've done all of our preclinical toxicology testing that is going to be necessary. We had a pre-IND meeting in the spring of this year, which was very successful. We're currently in protocol development and IND preparation. Our goal is to be in clinical trials sometime in early 2012. We want to submit the IND application hopefully beginning of December, but it may be more like January before we get there. So we've done toxicology testing in animals, we've done toxicokinetics testing in animals, we've done human tissue cross-reactivity studies in animals, all of which were very favorable, the results of which suggest that we have no reason that we can't proceed with our phase 1 study. We will be submitting further applications later in the year to fund phase 1B and phase 2 studies. We also discussed in general terms these studies with the FDA when we were there in April. They had some reservations about one of the studies that we proposed, but are willing to work with us on solving any issues that exist with the phase 1B study. The phase 2, they were they had no problems with our plans for our phase 2 studies. So again, we have a trial batch in vials. It's ready to go. The toxicology testing results were good. We're in the we're in the IND preparation phase. We're trying to finalize some of our analytical methods. Like I said, I'm already working with our contract research organization to set up our phase 1 studies. We anticipate the start of those phase 1 studies early next year, and we'll be submitting applications for funding for phase 1B and 2 studies toward the middle of next year. Where are we on our business development? We're still looking for funds for marketing study. We're going to continue to look for and develop relationships with companies that may either market this medicine or license it when it's ready to go. Also, we're always open for other opportunities and other chances to develop our medicines. Thank you for your attention. Do you have any questions?
Thanks, Brooks. Questions?
I'm just curious. You mentioned that the recidivism rate might be positively impacted because as they started up the methamphetamine again, that the receptors would keep it from being absorbed. Is it possible that because they weren't getting the buzz they were expecting that they would can they overdose with it?
Well, we don't think they can overdose with methamphetamine based on our studies in rats. We have attempted to kill rats, honestly, by giving huge doses. It's not just a one-to-one bind. There's more to it than that. It has to do with blood flow to the brain, the way the methamphetamine moves across the blood-brain barrier. But that is a concern that the FDA has, particularly with non-treatment seeking individuals. And so we're going to have to develop some toxicology studies showing that we can't overdose them. But yeah, absolutely, that's a concern. And it'll have to be something we'll have to do many risk mitigation strategies as part of our development. Yes, Jack.
How are you going to determine your dose of your antibody?
The first way is going to be in a phase 1B study where we can actually give a dose of the antibody followed by challenge doses of methamphetamine. You can do that in human studies. And so we will be able to determine, we'll do numerous doses based in part on our animal data, but also based on what we can give to humans based on other antibody studies. And then we'll start low and see where we can actually reduce the effects of an exogenously administered dose of methamphetamine. That's the only way, because of the endpoints that we're looking for in rats don't translate forward into humans. At least a rat can't tell you that, yeah, that felt good or it didn't. And so that's going to be the critical nature of the phase 1b studies in determining the ultimate efficacy of this medicine.
As an illegal drug, there's multiple formulations for how is there any way of preventing any kind of poisoning from different ingredients that are used in the meth? Because it's not always going to be the same ingredients that are used every time.
That's correct. This antibody works against amphetamine and ecstasy. It does not bind any other normal neurotransmitters. We will not be able to prevent every iteration that's out there, but the meth production techniques are actually pretty consistent. Now, is it going to prevent toxicity from an additive that's in it? No, it's not. But will it prevent the effects of meth? Yeah. I mean, and also the other area that we can't ignore and that we won't ignore is the opportunity for these users to go out and switch to another medicine. The FDA has told us that if they go and do that, then you can't write that down as a success. That's a failure, and we understand that. So we can't stop the end-stage user who is determined to use and overcome. What we can do is help the individual who really wants to work hard at trying to get over their addiction. And there are a number of people who are out, there's a fairly large percentage of meth users out there who are that way, who can't seem to get over the hump. But if they had a little bit of extra help, they could get over it. Brian.
You know, I was looking at your 4 ways in which you could approach this problem, and it occurs to me, and I know you're going to do a marketing study one of these days, and but the upper quadrant, the upper left-hand quadrant, where you talk about people who are there you go.
Mm-hmm.
Actually, it's the bottom right. Do you know how much money is spent on behavior modification as it relates to meth and ecstasy? Because wouldn't that be at least a starting point
That
to
would be
try
an
and
excellent
understand?
starting point.
And then,
Yeah.
of course, the question is, who pays for that?
Right. That would be.
That's part of what you need to find out for your marketing study.
Thank you. That's very good suggestion. I don't know the number.
And I guess the question is then, what do you think a marketing study that would really nail this down cost?
Well, our operations director tells me $150,000. Now, I don't know where she gets that number, but that's what she tells me.
Okay.
I don't know the answer to that.
And who's going to argue with Misty, right?
Nobody does argue with Misty.
He stole one of my questions, and it was about payment. But the second question, tell us again behavioral endpoints that you've looked at in the animal model. And I mean, I know you've done, all the physiology and the toxicokinetics, but just how do these animals act?
The simplest approach is to use a simple locomotor activity approach, where you give an animal meth, a rat in particular, and in 4 hours, a rat normally will move, say, 100 yards in a 4-hour period. If you give them a dose of meth that's equivalent to what a human meth user would use, they will move a quarter of a mile to a half a mile. So substantial increase in locomotor activity. We're seeing in some of our studies in the lab right now, we're seeing 75% reduction in that stimulatory activity in just that one measure. Another measure that we're using is a meth-primed self-administration model. So you get a rat will become addicted to methamphetamine. They'll punch a lever to give themselves methamphetamine IV. You let the effects of the meth wear off. You don't give them the meth for a period of time, and then you give them a shot of meth, and they'll start banging on the lever again, whether or not there's any meth being administered at that point. So in this model, our antibody prevents that meth-primed reinforcement, that meth-primed restarting of drug abuse by the rat. So again, do either of those translate into what a human is going to feel and how a human determines whether or not they're going to go out and use meth? We don't know. And the FDA and other people who've studied vaccines for other drugs of abuse, cocaine and nicotine, for instance, suggest that the only way you're going to figure this out is to go give it to humans, give them some meth, and see what happens. And that sounds like fairly base, but that's the only way that we're really going to be able to tell. The last way is pharmacokinetically. We can measure brain concentrations of meth in rats after we've given them the antibody, and they are substantially reduced, if not brought to zero, soon after administration of the antibody. And so we see these pharmacokinetic changes. That, if we see that, that's something that will be consistent in humans. We know that given the way antibodies work, they're going to increase blood concentrations. They're going to get it out of its sites of action. So those are the three primary methods that we're using to determine to predict efficacy in humans.
I want to ask about the stoichiometry of that. You got an antibody which weighs over 100,000. You got a drug that weighs 200, and so if I don't know what the dose of methamphetamine is, but the looks like the number of molecules of
That's correct.
Of antibody that you got to give to equal the number of.
You don't have to equal. That's the point. With the PCP antibody, for instance, we showed that 1/100th at most 1/100th the dose binding sites of antibody was effective in reducing that dose of PCP. So it was 100-fold less amount of antibody to effectively change the dose of PCP, the effects of PCP in the brain. It's more like the antibody has a we'll talk science in a minute, has a very rapid on and off rate. What we think is happening is that the antibody is high enough affinity that it draws it out of the brain, but then it goes back off of the antibody in the bloodstream, but now it's moved out of the brain and back out into the bloodstream where it can go to inert tissues. The antibody circulates back into the brain, it can get another molecule, take it off. So it's more like a magnet getting iron shavings. You know how it doesn't have to get every last one of them attached to it to make some of them move. It can pull a trail of iron shavings along behind it. And that's kind of what the antibody appears to be doing, because even the cocaine data with the cocaine active vaccine, where they saw in patients who got titers, significant titers, they had substantial excesses of cocaine on board compared to the amount of antibody that was on board, and it still reduced the effect of cocaine. So it's not just a one-to-one binding. You don't have to have one molecule of antibody or one for every 2 molecules of methamphetamine. It has to do, again, with the fact that 20% of blood flow goes to the brain in humans, the fact that the blood-brain barrier there is no barrier to moving a methamphetamine into and out of the brain, and the fact that we're setting up an equilibrium with the antibody that gets it back out into the bloodstream and into areas where it can't be as harmful.
Brooks, one last question. The National Institutes of Drug Abuse have been incredibly supportive of this work. I mean, you guys are the poster child of sort of what they've been doing. Mm-hmm. Sort of relative to the question that Dr. Clevenger was asking, what do these guys think about sort of the market issues, and where are they sort of pulling a lot of their justification aside from the good science to really get behind this effort? Do they talk about that at all?
Well, I think that the fact that they have nothing else at this point is one. They're looking for any new technology to get into humans and see where it goes. We may learn a lot we may be able to find out that we can make this even better much less expensively just by getting into humans and finding out how it really works. I don't think they're as concerned, at least initially, about the market as they are about just getting it out there and getting it to try and getting it tried in humans. We have asked them for some help on this. They said, sure, we'll give you some, apply for a grant.
Yeah, we're here from the government. Yeah. We're here to help you, right?
Yeah. Yeah.
Okay, all right. We've got a lot of time here until 3 o'clock, so we're going to go ahead and break, and then we'll have our break at 3 o'clock. What we might try to do is get started just a little early so we can give one of our shorter talks a little bit more time. So let's just say we'll be back in here 5 after 3. We'll kind of split it down the middle, and continue on with our afternoon session, which will be very, very strong, and look forward to seeing you all here. Thank you very much.